I go to work every day with a good feeling because as I've been part of the development team, this was years and years of planning and developing, and now it's actually there. So I'm very excited to actually use these to the maximum potential. I'm Tom Theeuwen. I'm a researcher at Laboratory of Genetics, which is part of Wageningen University and Research. I'm a geneticist and I study the genetics underlying photosynthesis. And measuring photosynthesis is a very complicated trait. It takes a lot of time to measure a few plants. And to do genetics, we need hundreds and thousands of plants to be measured in a short time frame. So for that reason, we've built these facilities where we can measure hundreds or thousands of plants within an hour and get very good, reliable data. I'm currently working with the model species Arabidopsis thaliana, which is sort of the model for plant sciences in general. In this particular case, I grow a collection of accessions from Ireland. We're very curious to see whether there's accessions that respond very different to typical environmental conditions. Some will probably respond happily, others will respond very dramatic, and I'm looking to find out what is causing these differences. So what is the genetics underlying these differences? I was indeed involved in the development of these facilities. As an experienced user already before, I still had the desires to use bigger facilities with better environmental conditions, with better cameras. And that's why I was involved, to really see what is really pushing the limit of what is possible within the current technology. Yeah, we have here 4 different climate chambers, and together they can mimic effectively any environmental condition globally. So that allows us to study any plant within any environment and how it responds to that. Every room has an area where plants grow, and we have designed them so that they can receive exactly the same light conditions, temperatures, CO2, etc. And that ensures us that they respond the same when we measure them. Yeah, the type of research that I'm doing will effectively not be possible without such facilities. I just need the throughput, so I need to have thousands and thousands of images within a very short time frame. Otherwise, the environment has already changed and I don't know the response of all the same plants. So I can't do my research without these facilities. A G8 is really designed to produce very high light intensities and grow a lot of different Arabidopsis plants in there. And in this condition, we are able to mimic very high sunlight and then measure how these plants respond to that. And simultaneously, we can measure chlorophyll fluorescence, thermal imaging, and hyperspectral imaging on all of these plants in roughly an hour. And that gives us very detailed information about how all of these plants respond to these high light conditions. That's what we see here. They're really state-of-the-art facilities that allow us to do maximum research at the level we really need to be done.